Hello, everyone. I have made this recording for you to help you prepare for the ASBMB certification exam for those of you who were not able to attend the help session tonight. I'm just going to click on this link right here, and I'll leave it here so that you can see it, so that you can maybe try and Google it. I did actually send you a copy of it. So these are the accreditation exam sample questions. So they were posted a number of years ago, but that is what we are getting in terms of having an idea of what kinds of questions that you will get. So if you go to the asbmb.org website and just navigate through education and certification exam, you should be able to come up with this link. So I'm going to go through each of these questions, and starting for the first one, so the concept area is energy and metabolism. So we are given our favorite reaction, a malate dehydrogenase, and we see we have a very large positive delta G. And this final reaction, so that we're told the final reaction of the citric acid cycle has a standard free energy change of 29.7 kilojoules per mole. So that you would think that that would be unfavorable. So how does this in fact occur in the cell? So what we should emphasize in this exam is that big concepts are really important. And there are lots of details in the questions, but you're given the details. You're told it's the citric acid cycle. You know that it's a cycle. You're told it's the last reaction. And because it's the last reaction in a cycle, it will be followed by the first reaction, where you bring in another molecule of acetyl-CoA to condense with OAA. So this reaction in the malate to OAA direction, if you try to do it in a test tube, it would not go that way. You'd have to add OAA and have it form malate. That's the way you would have to do it. But in the cell, the reaction does in fact happen this way because a number of reasons. One is that the product of this reaction serves as the reactant for the next reaction. So as you're constantly removing product, you're using it as a reactant. So if you constantly remove product, that's going to help you move the reaction in this direction. The other thing we should think about is that there are actually two products. NADH is one of them, and since this is the citric acid cycle and we are in the mitochondrial matrix, that NADH is going to go off to electron transport. So you're removing that product too. So we have two products, and we have mechanisms for removing both of them. The other thing is when we go back and we look at that positive delta G 0 primed, again, notice that that is a standard delta G, and that would be if we had 1 molar concentration of reactants in the cell, which we do not have. So we use the delta G. So the delta G is the criterion for spontaneity, and this is 0, which it means maybe you could go either way depending on the conditions, but it's certainly not that really large positive value. So what we need to emphasize here is when you're given a question like this on the exam, we need to think of big principles. So what kinds of things? So again, I just copied this from our Biochem 1 class. So we use this to figure out what reactions would be regulated, and malate dehydrogenase isn't one of them, but that isn't the point. The point is, what strategies can we use to drive a reaction forward? So driving a reaction forward that's not favorable is also a concept followed here. So again, trying and think of how I'm going to drive a reaction forward. So the reaction that's given is, if you look at, I can go back here to the exam, is this exam right here is given by the UDP glucose pyrophosphorylase. So if I go back to my slideshow here, just to put this in context, that is this part of the glycogen synthesis pathway. So you take glucose 6-phosphate, isomerize it to glucose 1-phosphate, and we're going to want to take those glucoses and string them together to form glycogen, but you can't just string together glucoses to form glycogen. It's not energetically favorable. So we make something like, maybe we could call this activated glucose, where we use UDP glucose, which ends up being the substrate for the glycogen synthase. So this picture is from our book, and it's almost identical to the one that's given on the exam. So again, there's a lot of detail here that is presented that you don't have to know, but you do have to be able to interpret. So we have glucose 6-phosphate as isomerized to glucose 1-phosphate. And here we're shown that there's a nucleophilic attack of this phosphate, and then this pyrophosphate is the leaving group. And what we do is we hydrolyze a phosphoanhydride bond on UTP. So it's similar to ATP. So we hydrolyze that bond, and then we get this activated glucose. So what happens here is that we end up hydrolyzing another bond here. The pyrophosphate was the leaving group, but we're going to hydrolyze this phosphoanhydride bond too to convert the pyrophosphates to regular phosphates. We end up synthesizing a phosphoanhydride bond too. So we have the net hydrolysis of one phosphoanhydride bond. So in this question, they say, why is this step necessary? Well, that step is necessary because if I only was going to hydrolyze one and form one phosphoanhydride bond, that's not going to be energetically favorable. But if I hydrolyze two phosphoanhydride bonds when I form one, that's favorable. And so that's the phosphoanhydride bond that I formed. These delta G values are taken from our textbook. So here, this shows the first reaction. So what the question is asking is like, why do I have to do the second step? Why can't I stop at the first step? Well, in the first step, you both hydrolyze and form a new phosphoanhydride bond. And what you have to do then is you need the hydrolysis of a second phosphoanhydride bond. And that's that second reaction right there. And this second reaction is catalyzed by the inorganic pyrophosphatase. If you add these together, what you're doing is you're coupling two reactions together. So that's another strategy, is to couple two reactions together so you can make the overall process be favorable. Okay, if we go to the next question. Okay, so I'll go to the next question. Let me go back to my browser. Okay, so here we are given the macromolecular structure function regulation. And I hope that when you can see this description right here, that the hydrophobic effect is what you should be thinking is going on here. So the correct choices are A, C, D, and F. So phospholipid bilayers, protein folding, and folding of transfer RNA. So I have pictures here that I can show you. So amphipathic lipids form bilayers. So we have the polar head groups very favorably interacting with water. Water refuses to interact though with the nonpolar chains. It doesn't do so very favorably, so the nonpolar chains are forced to interact with each other via the weak London dispersion forces. So amphipathic lipids form bilayers as a consequence of the hydrophobic effect. And that's shown here. My cells do too, but my cells have one tail, so they form sort of this conical structure. And this shows how we form the lipid bilayer. So again, favorable interactions between water and the head groups, which are polar and charged. But these long hydrocarbon tails, water does not form favorable interactions with those, so that forces them on the inside. So the same thing happens with protein folding. If we look at this diagram right here, where if we imagine a protein that's unfolded, and if we color code the most hydrophilic residues blue, like water, most hydrophobic yellow, like oil, and then something in between, if we show how we fold these up and then we just kind of peel away, we can see that inside are the oiliest, the most hydrophobic residues. So then, again, this is the consequence of the hydrophobic effect. So I have I got this from a French website, and they forgot to turn off their spell checkers and their paragraph numbers, but I still like it anyway. So this shows that folding a protein doesn't, in fact, violate the second law of thermodynamics, and that we form a highly folded protein because the water molecules associated with the protein are very, very disordered. If you tried to make the water molecules interact with the hydrophobic groups, they wouldn't do so very favorably, and they might be forced to form these ordered cages, which is not favorable. So we would have to look at the whole system. What about the next answer, which is DNA? So DNA, we have a sugar phosphate background, very polar with the sugars. Phosphates, very polar and charged. And in the center, we have fairly nonpolar bases, which interact with each other via very strong stacking interactions. Hydrogen bonds are important there too, but the stacking interactions are the most important. And the last answer to this question, dealing with tRNA, what's interesting about transfer RNA is that you can really look at it kind of like protein structure. So if I just look at the structure of this, this primary sequence of the tRNA, I can fold my transfer RNA into these stem loop structures. So maybe I consider that to be secondary structure. But the overall tertiary structure of this tRNA looks like this, and it folds the way proteins fold, consistent with the hydrophobic effect. And you do have to have the anticodon be accessible so that this can be read by the messenger RNA at the ribosome. But the folding of this tertiary structure is also the consequence of the hydrophobic effect. So when I looked at all of the answers to this question, I was really struck by the formation of the structures was really consistent with the hydrophobic effect, in which polar groups interact favorably with water, but water refuses to interact with nonpolar groups. That forces them to the inside of a structure where they are not available to interact with water. Of course, we have lots of other kinds of intermolecular forces involved, like hydrogen bonding and dipole-dipole interactions. Salt bridges or ion pairs that are also important. So all of these structures are important in determining structure of whether it's a protein, DNA, a tRNA, or a phospholipid bilayer. Okay, this next question, again, I'm going to go back to my diagram here. So this has a diagram of so this is the pyruvate dehydrogenase complex. So if again, read the question really carefully. Dehydrogenase, already we know that means that's a redox reaction, because dehydrogenases catalyze redox reactions. So it says the prosthetic factor lipoamide is attached to dihydrolipoyl transacetylase through a lysine, and then they show the reactions. And the question, which is bold, is what chemical entity or entities does lipoamide transfer or carry going from step 3 to step 4? I decided to use this picture because I liked it better. But the same step is right here. So the lysine is attached to this lipoamide group, and you can see right here that this ring is open and these sulfurs have hydrogens bonded to them, and there will be electrons, 2 electrons shared between the sulfur and the hydrogen. And when you go on to the next step, hydrogens and electrons are donated to FAD to form FADH2, and the consequence of this is that we close the ring. So hopefully you can see that the answer to this question is very simple. We have 2 protons and 2 electrons. And I think if you examine this really carefully, even if you didn't remember this mechanism at all, you could see that we're losing 2 protons because the protons are ending up here. But hopefully you can also see that if I have 2 electrons here and 2 electrons here, that's 4, but I'm forming a bond here, so that means I'm using 2. So that means that I must have 2 electrons. You might not remember that if you look at FAD, that when you look at this ring, that the FAD, when it becomes FADH2, I think it's obvious that that we have 2 protons, but we also have 2 electrons that are also forming these bonds between the nitrogens and the hydrogens. So again, these are dehydrogenases, so we know that they're redox reactions. So when we're thinking about our answer, we should be thinking in terms of redox reactions and the kinds of things that are transferred. Okay, so the next question wants to know about why the boiling points are so different. So hopefully that you can see that you have a hydroxyl group right there, and hydroxyl groups are really great at forming hydrogen bonds with their neighbors. So we have a hydrogen bond donor here. This H is a hydrogen bond donor. This oxygen is a hydrogen bond acceptor. We have hydrogen bond acceptors over here. Here we have hydrogen bond acceptors, but no hydrogen bond donors. And also, in terms of reading the question very, very carefully, we should note that we are given the information that we have same chemical formula, same molar mass, boiling points are different, and says solely and entirely 100%. So they're giving us a lot of clues that you just have these liquids and nothing else. These are not dissolved in water. We're biochemists and we tend to think that way, but these are 100% these liquids. So this is something maybe you would have learned in general, or maybe organic chemistry. So these are not dissolved in water, so your answer shouldn't include any kinds of interactions of these compounds with water. What you'd have is you'd have interactions of these compounds with each other. So again, we have hydrogen bonding as a possibility right here. Here we have London dispersion forces and dipole-dipole interactions, which are not as strong. So if your intermolecular forces between these molecules and your neighbors are not as strong, then it doesn't take a whole lot of heat energy to break them, to convert the liquid into a gas. You'd have to break these intermolecular forces, so you go from a liquid to a gas. But if the interactions are very strong, it's going to take a whole lot more heat energy to break those intermolecular forces to convert the liquid into a gas. So again, here's my list of intermolecular forces that we talked about in the course. So we had ionic interactions, which are not shown here. Van der Waals forces, some books just lump hydrogen bonds, dipole-dipole interactions, and London dispersion forces just all into a collective Van der Waals forces, which is probably why you shouldn't use that term. It's not very specific. So you should use terms like hydrogen bonds, dipole-dipole interactions, and London dispersion forces. So in this compound right here, we have the possibility of all three: hydrogen bonds, dipole-dipole interactions, and London dispersion forces, whereas the compound that was on the left, all we have are dipole-dipole and London dispersion forces. And again, you can see by the bond strength that they're not nearly as strong as the hydrogen bonds. Okay, so those are our favorite IMFs. All right, the next question had us looking at deamination of cytosine to uridine. So again, this isn't something, maybe you didn't know about this, that if we do a hydrolysis that we convert cytosine to uridine. But what we're asked in the question is, suppose we had an asparagine, and they give us the genetic code, we don't have to memorize it. Suppose we have an asparagine, and they give us the 2 codons, and what would happen if there was a cytosine deamination that occurred in the codon? Now, they don't tell us what it is, but it's got to be this one, the AAC, because it's the only one with a C. You can see if the C is deaminated to the U, we still have asparagine, so nothing happens. We have a silent mutation, so the protein should still function as normal. But the second part of the question says, what if we have glutamine? So, and they don't tell us which codon is which, so we should answer for both. So if it's CAG and you go to a UAG, you get to a stop codon. But if it's CAA and the same thing happens, you go to UAA, you also get a stop codon. So the answer is the same either way. No matter which codon with glutamine you're talking about, because they don't tell you. So you would get a truncated protein. So the translation will stop when it's not supposed to. It'll stop early. So you get this shorter truncated protein. The protein will certainly probably not function properly. It might even be in danger of not folding properly, which means it could possibly precipitate. So that would trigger the cell to degrade the protein. So you might not have any protein at all. You might initially produce a truncated protein that will be degraded. So the question 7 is, I'm going to go back to this. It's rather lengthy, and I'm going to let you read this on your own. Okay, so we already did that question. I'm going to let you read this on your own. So you can stop the tape right now if you don't have the document, and if you want to read this on your own. But basically, what this comes down to is that there are different ways that mutations can occur. So we're being asked why the mutant gene was not passed on to the progeny. So we're actually even given the answer. We just have to explain why. We're not even being asked to explain what happened. We're being told that the mutation's not passed on to progeny. So what that means is, if the mutation's not passed on to progeny, then it must be a somatic mutation. So I found this table online. I don't completely like it because there are a lot of typos, but that means that the mutation is in some other cell, not the germline. And because it's not a germline, it's not going to be passed on to progeny. So in the narrative, you can see that it was acquired somehow. It could occur in any body part at any stage of a person's life. And maybe it's present in cells such as kidney, liver, or skin. It's going to be localized, and it's not doesn't have a definite role in evolution. So you can, if you read that scenario in the question, this totally lines up with that. Whereas a germline mutation would be in either the sperm or the egg, which means that you're going to pass it on to progeny. That happens sporadically, but that's going to affect the entire organism, and that happens during germline division. And it will be presented in all cells. And if you read the scenario, that's definitely not what's being discovered. Okay, so I found another picture on the same website. So again, if you've already are born and there's some kind of mutation after you've already been born, then you have some kind of somatic mutation. Whereas if there's a mutation in either the egg, so here it's shown in the egg, actually I don't know where no, I don't think it specifies. If it's in the egg or the sperm cell, then your mutation is the embryo, and that's a germline mutation that you'll find in the entire body. Okay, so the next question has to do with information storage and transfer, and here I have all of the answers. So this expects you to have some kind of understanding of the DNA makes RNA makes protein, and there could be some strategies that could help you with, do these things happen transcriptionally when DNA makes RNA, or translationally when RNA makes protein, or after the transcription and before translation or after translation. Some of these processes, just to use a little bit of strategy, if you haven't heard of them, you might be able to figure out from the term. So for example, messenger RNA splicing, well, that has to be post-transcriptional because you already have your RNA. And a repressor is going to determine whether you make your RNA or not. So that's going to be transcriptional. A signal peptidase, well, if I'm acting on a peptide, I must already have my peptide. So that must be post-translational. This is RNAi or RNA interference. Again, I've already have my RNA, so that must be post-transcriptional. Messenger RNA deadenylases, again, I already have my messenger RNA, so must be post-transcriptional. And of course, ribosome is where the translation happens. So you might be able to answer this just by doing some deductive reasoning. But I just have my favorite pictures here. So I have DNA makes RNA as a primary transcript, and then you have capping, splicing, and polyadenylation. All of this RNA processing happens after or post-transcription. And then I have my messenger RNA, which is exported into the cell, and then it encounters the ribosome, and it is translated into protein. So that's not as detailed. Here, I thought I would put the lac operon because it's an example that a lot of people know about. If you have a repressor that binds to the operator, which is part of the promoter, that prevents transcription. So you check the transcription box because this is a regulation of transcription. The other thing about the deadenylase is that you might remember that messenger RNA has these really long poly A tails, and if the poly A tails get to be too short, then the messenger RNA gets degraded. So again, if the messenger RNA is already made and it's being degraded, that's happening post-transcription, but before translation. And this is a nice diagram of the whole system. So here I have my my messenger RNA, and if it's a cytosolic protein, that it stays in the cytosol, it might be targeted to peroxisomes or chloroplasts or mitochondria, or the ribosome could happily be making the protein, and maybe an endoplasmic reticulum, an ER signal sequence emerges, which will direct the ribosome to the endoplasmic reticulum, and there's a pore through which translation continues, and there will be a signal peptidase, so that's where that comes from, that will remove the signal sequence. Once you're already in the ER, you no longer need that sequence, so it's removed. And this protein can then continue on the secretory pathway. So maybe it stays in the ER, maybe it stays in the Golgi, maybe it goes to the plasma membrane, maybe it goes to the lysosome, or maybe it's completely secreted from the cell. So having a good general idea of how DNA makes RNA makes protein is a good strategy for doing well in this exam. So the next question gave you sort of a hypothetical, okay, so we already did that one, gave you a hypothetical novel amino acid called antarctine. So I decided that I was more interested in real enzymes. So I went and looked up the sequence for these branched chain amino acids. So if we look at, say, leucine, so we have a number of steps that we take to get to leucine, and all of these are synthesized when you go from reactant to product. These are all catalyzed by enzymes. So if any one of these enzymes is not working, you will not make leucine, and that means your bacteria, you will have to give them leucine or they will not grow. So if we go back to our hypothetical question, if I go back here to our antarctine, we are told that we are looking at a plasmid, and the plasmid carries the we want to rescue this mutation, so that we found a mutation and we're going to rescue it by encoding the proper enzyme on the plasmid to see if we can rescue this, but we can't. So it's a long scenario, but what we're being asked is, what are some of the reasons why? Well, maybe there was another mutation. So remember, it's a pretty long pathway. Maybe there's more than one enzyme that's affected. So it affects maybe not just that gene, but other genes. What you're supposed to be doing here is to come up with scenarios that can explain the results that you see. All right, so let's go on. So the next question then is, all right, let me go on, is you are given the structure of histidine. This is not how I would draw histidine, but anyway, you're given the histidine with its pKa's. So the alpha carboxylate group is here. The side chain is this imidazole ring here. And here is our amino group right here. And then we're asked to figure out the charge when dissolved in buffer. So we're giving pH 4, 7.5, and 11. So if we go back here, I drew these out. So what I did is I drew my amino acid completely protonated. So the amino group's protonated, the carboxylate's protonated, and the ring is protonated. So if I raise this to pH 4, that's a pH greater than this pKa. So this proton's going to come off, but these protons are going to stay on because the pH is less than the pKa. So that's going to give you this structure right here. So this cancels out, so the charge is +1. If you then raise the pH to 7.5, now this proton is long gone. This proton is going to dissociate too. Again, the pKa's are less than the pH, so this proton will still be here. So now I have the Zwitter ion, so that has no charge. And then if you raise the pH to 11, all three protons dissociate because the pH is greater than all three of the pKa's. And now we have an amino acid that's a -1 charge. So it's highly likely you'll have some kind of question, something like this on the exam. Okay, so this question, I think this is the last question, is scientific method and quantitative reasoning. So you're given a scenario, and again, you're not really expected to know anything about this. You're given a scenario, and then you're asked to come up with hypotheses. So if you can read the scenario on your own, but maybe a hypothesis is that expression of the reporter gene requires T beta but not T alpha. Or maybe expression of the reporter gene requires the presence of both T alpha and T beta. So you would start with the hypothesis, and then you would do this experiment to either support or refute the hypothesis. So you're not even asked to do this. You're just asked to come up with two hypotheses that you could use. And then this is the experiment that that you would use to support or refute the hypotheses. So the notes that I have say that these have to be expressed as sentences and not as questions. And if you express this as a question, it will be marked as not yet proficient. That's a very nice way of saying it will be marked wrong. So if you're asked to formulate a hypothesis, do not do it in the form of a question. Okay, so other questions you might expect, just so to wrap up here, just from other, looking at all of the objectives that the ASBMB has for its certification program. Of course, we're going to have some kinds of general chemistry tests. You might be asked to do some kind of like solution calculations, grams to moles, molarity, percentage, things like that. You might need to recognize different isomers because that's important in biochemistry as well. And we really didn't talk too much about enzymes. So you might be asked to look at different the types of catalysis or catalytic strategies. Is acid-base catalysis. So acids donate protons, bases accept them. Covalent catalysis, that means you have a substrate or portion of the substrate temporarily covalently bound to the enzyme. You should learn how to calculate Km. So if you're given a Lineweaver-Burke plot, that's the negative of the x-intercept. For Vmax, it's the reciprocal of the y-intercept, and you should be able to, by inspection, look at various different types of inhibitors, competitive, uncompetitive, non-competitive, and be able to, by inspection, discover the type of inhibition and be able to say what happens to the Km and the Vmax. Do they change? Does one of them change? Do they both change? Do they go up? Do they go down? That's something else you should know. So you should look that up. And you might be given a graph and be expected to read the graph. So that is it for my help session. I hope that you found this helpful. So my advice would be to very slowly and very carefully read the questions. A lot of detail is present in the question that you need to pay attention to, but that you don't necessarily need to know. You just need to come up with the overarching principles that you've been studying all the way throughout your college career. So I think that you will do very well on this, and good luck.